Thank you so much, Tara. It's a pleasure to be here. Let me know if you can't hear me in the back, I think. But yeah, it's great to be here to come and talk to you guys about our research. I apologize in advance, I didn't simplify these slides very much. So hopefully things still make sense, and if they don't, please yell out and stop me from using all the jargon that we usually use in the lab. But today I'm just going to tell you a little bit more about the human microbiome, in particular the trillions of bacteria that live in our distal gut. And so we like to say that we never dine alone. So even if you don't have any human friends, every time you sit down to have a bowl of Cheerios, you're joined by these trillions of bacteria in your gut and archaea. And they help us do a lot of different things. And so I'm just going to talk about one particular role that they have, and that's influencing how we process our diet. Okay, so just as a brief background, as Curtis pointed out, the human body actually has 10 times more microbial cells than human. And so if you just go in total, the amount of human cells, we're really outnumbered by these microbes, and that includes microbes from all three major domains of life. There's small eukaryotes, there's archaea, there's bacteria, and there's many different viruses as well in these communities. And the largest collection of these microbes is actually in our gut, so that's what we typically call the gut microbiota. These microbes also have a tremendous diversity of genes, so there's an estimated 100 or maybe even higher fold more genes in these microbial genomes than in our own human genome, and that's what I'll be calling the microbiome. And some early studies really showed that the gut microbiota is composed of 10 major groups of bacteria. So we typically refer to them as phyla. They have different names, like the Bacteroidetes and Firmicutes, which are the two most abundant groups of these bacteria. But there are another 8 less abundant groups. And due to some research, which I'll share with you some details a little later in the talk, we're starting to get a picture that each person harbors a unique collection of these microbes in each habitat. So if you compare samples taken from the gut of many different people, we're each sort of colonized by our own unique fingerprint of these bacteria. Okay. So this community actually contributes a lot of different metabolic functions that we don't normally have. And one of them that we think is particularly relevant is how they act upon our diet, as well as some carbohydrates that are shed from our own human cells. And so this is just like a really rough depiction of what the gut might look like. Obviously, it's much more snaky than this, but this is sort of a straight gut. And you can imagine you're eating at the top of your gut monosaccharides, things like glucose or fructose, maybe disaccharides like sucrose, but then also much more complex polysaccharides that are chains of glucose or other simple sugars connected together. And then our own body is really good at taking these monosaccharides and rapidly absorbing them in the small intestine, and then that contributes to our energy balance, but we're relatively bad at accessing these much more complex carbohydrates. And so these make their way down to the distal gut, or the colon, where they serve as a substrate for these trillions of different bacteria that are living there. And these bacteria use a process called anaerobic fermentation, and they take these complex carbohydrates and spit out something called short-chain fatty acids. So this is just a waste product of these bacteria that is then absorbed by the host and used as an additional energy source.
Wait, this is the colon?
Yeah,
so there's absorption from the colon.
Exactly, yeah. So the colon is sort of this region. Obviously it doesn't look like this, but this is this is just a schematic to sort of show you the proximal small intestine, the distal, and then the colon at the end, yeah. And there's various estimates as to, how important this may be and what fraction of the calories that we take in are actually coming from these short-chain fatty acids. So just in a broader sense, I want to emphasize that we're really living in a microbial world. So if we look at the evolution of life on the planet, we can do something called 16S ribosomal RNA gene sequencing. That is a particular gene that allows us to basically barcode different organisms and create a tree of life. And so this is sort of an older version of the tree of life from a couple decades ago now, where they showed that there's three major groups, the bacteria, the archaea, and the eukarya. And if you just zoom in on where the humans are and where all these large organisms that we're usually studying, like corn or dogs or mice, that's a tiny little portion of this tree. And so almost all of this diversity is in the microbial world. Additionally, it's really largely uncharacterized. So these are just the broadest group of bacteria, so the bacterial phyla. In 1987, we knew 12 of these phyla. Now, this is the latest count in 2006. There's 100 different ones that have been described. And then the blue are just the ones where we even have a single cell that's been cultured from these broad bacterial groups. And just as a sense of context, the amount of diversity within a single phyla of bacteria is basically as large as the distance between a human and a tree or a human and yeast. So these are tremendous differences. And so in 2006, we stand where only 30 of these even have a single representative, and 70 of these 100 different phyla are completely uncultured. And even in the ones where we have a representative that's been cultured in the lab, we may only have one or two. So we're really only sampling a small amount of the diversity in culture. And so that led to the development of these metagenomic approaches. And so this is just a general sort of schematic of how these types of studies go on. You go to your environment of choice. So a lot of people might go to the ocean on their yacht, or they might go to a soil sample. They might go to a hot spring or somewhere exciting. We would just go and sample someone's colon or feces. But that's much more exciting to us. And then you can just extract all the DNA that's in your sample, or even RNA or proteins or metabolites. And typically for DNA or RNA, then you might clone it into, we call it making a metagenomic library, typically in a host like E. coli. Or now you can just bypass this step and directly sequence the DNA or the RNA. And that allows us to do a variety of different types of computational biology analyses on these data. But it really allows us to access the genomes of these organisms without having to actually culture them. Okay, so the final sort of technical thing I want to point out is that a lot of the studies I'm going to tell you about rely on the use of a technology called Gnotobiotics. And so this was really a pioneering technology that was developed in 1950. And what it allows is that you can create these large vinyl isolators where you can raise a mouse from birth to death without any exposure to bacteria. And so this is a really unique tool because basically all mammals are colonized as soon as they're born and then remain colonized throughout the rest of their life. And so this allows you to make what we call germ-free mice. And then we can compare germ-free animals to conventionally raised animals that live their entire life with exposure to bacteria. And additionally, we can make what we call conventionalized animals, where we start with a germ-free mouse, and then we take particular bacteria that we're interested in studying, or maybe a whole community of different bacteria, and colonize these mice and see what happens. Okay, so the first experiments that were done by Frederick Backhead, who was a postdoc in Jeffrey Gordon's lab in St. Louis, just wanted to compare these three different types of mice and see if there was any difference related to body weight or obesity. And so what he did was just look at the total body fat that was in the bodies of these animals in germ-free mice versus mice that were recently colonized, so just 2 weeks after they were colonized by microbes, or conventionally raised animals that had been colonized ever since they were babies. And what he showed was that you see this increase in total body fat in these animals. And the perplexing thing was that these animals are actually eating less food. And so they're taking in less chow on a day-to-day basis, but they're somehow gaining much more body fat. And this happens in male and female mice, and it happens in many different types of mice. And that sort of raised the idea that maybe these microbes might be actually contributing to body weight. Or to fat. So we followed this up with the study of these two different mouse models of obesity. These are relatively well-characterized mice. The first is an interesting mouse line called Obiobi mice. And these mice, which are shown on the right, really develop a tremendous amount of obesity. So you can see this is a sibling that's lean. And then mice with this mutation in the Obiobi gene become incredibly obese. And the reason this happens is because they don't express one particular protein called leptin that causes them to really dramatically overeat, as well as impacts other parts of their physiology. And then there's another mouse model of obesity where you just take mice that are all exactly the same, so they have the same genes, but you feed one a low-fat, sort of healthy diet, and you feed the other one a high-fat, high-sugar diet, which we call the Western diet. So we're forcing these mice to go to McDonald's every day for their entire life, and they also become quite obese. But we wanted to just ask whether or not there were different microbes found in the gut of these obese mice versus lean mice, see if there's some correlation between obesity and the microbes that are in your gut. And so in the bottom left is a graph just showing the two major groups in the gut, the firmicutes and the Bacteroidetes. And you can see if we compare lean, either wild type or heterozygous mice, to their obese litter mates, there's this shift in the relative abundance of these two major groups, where the firmicutes increase in abundance, the Bacteroidetes decrease. Similarly, when we transfer the mice from a low-fat diet to their high-fat McDonald's diet, we again see an increase in the firmicutes and a decrease in Bacteroidetes. So this sort of indicated to us that maybe there's some association here. But we wanted to design some sort of experiment to see if these altered communities could actually be contributing to obesity in these animals. And so we did what we call a microbiota transplantation experiment. So this is sort of a strange design, I think, but allows us to maybe start to get at causation and hopefully in the future start teasing apart the mechanism for these things. And so the way we do this is we just start with a huge panel of germ-free adult recipient mice. So these mice are just waiting around for some microbes. And then we take different adult donor mice, where one donor might be lean and one might be obese. And this might be either due to the Obiobi mutation or due to a change in diet. And then we just colonize these recipient animals, wait for a while, and check to see whether or not there was some impact of this community. And so the first thing we looked at was just whether or not these mice increased body fat after they were colonized. And so on the left, if you look at colonization with samples taken from either a lean mouse or an Obiobi mouse, you can see that 2 weeks after this colonization, a mouse given a sample from a lean donor might gain about 25% body fat, but then mice given a sample from an obese donor actually gain about 47% body fat. So there's this increased amount of fat gain due to this different inoculating community. And this also happens in the case of diet-induced obesity. And these differences weren't really attributable to the amount of food that these mice were eating. So there was no difference in the amount of chow they were taking in. They all sort of started at the same initial body fat and initial weight. And so it really seemed to indicate to us that these different communities might be actually contributing to obesity. Okay, so next we turned to humans to just see whether or not any of the things we were finding in mice might actually hold up in a human study. And so at the time, this was a relatively small cohort. So we recruited 12 people that were part of a much larger comparison of low-fat versus low-carbohydrate diets. And then we just looked at them throughout a year. And so this was actually done by taking fecal samples. So we'd take an initial sample before they went on their diet, a sample 12 weeks later, and 26 weeks later, and then 52 weeks later. So we took these 4 different time points throughout the year, and then just looked at the relative abundance of these 2 major groups in the gut. And so hopefully you can see from these graphs that over time, the average amount of Bacteroidetes are sort of increasing in these individuals. And on the bottom, you can just directly correlate these 2 things. So you can look at the change in Bacteroidetes, so how much Bacteroidetes was gained versus the amount of weight that these people lost. And so there was this correlation between losing weight and how much Bacteroidetes was left in the gut, indicating that maybe there's some similar things happening in our mouse models to what's going on in humans.
But did they lose weight or did they lose fat? Or both?
Yeah, unfortunately only had, we were only looking at a change in weight, so not quite as good as in the mouse experiments where we're
So they
actually
lost
looking
weight.
at, yeah. And so we followed this up with a larger study where we were interested in some additional questions related to lean versus obese, but also related to just whether or not families might share similar community structure and whether or not that might be determined by how closely related you were to someone in your human genes, so whether or not you were an identical or a fraternal twin. And so we got samples from monozygotic as well as dizygotic twin pairs and the mothers of these twins, and this was a total of 150 people. And then just took an initial sample, but then also a second sample 2 months later. And we analyzed this using 16S, where we were sort of asking the question of which bacteria were in the samples, but also shotgun sequencing the DNA just to ask what genes were there and what genes change in abundance in lean versus obese. So this is going to be totally technical, but well, maybe I'll just skip this. I guess the main point is that we have a method that allows us to just take the profile of bacteria that we find in each sample, and we can compare and ask for a different type of comparison, whether or not things are more similar or more different. And so this is the y-axis of this bar graph, where the higher we are, the more different a given comparison is, and the lower, the more similar. And so on the left and the right of this bar graph, you can see that if we compare unrelated people, they're the most different from each other of all the groups we're comparing. Whereas if you look at a person at the initial time point, but then 2 months later, they're much more similar than these unrelated people. And in the middle, we have comparisons of twins to their co-twin and also twins to their mother. And in both cases, these comparisons are much more similar. And so there's some similarity between a given twin and their co-twin, but also a twin to their mom. And sort of surprisingly, we expected to see that monozygotic twins would be much more similar than dizygotic twins because they are identical in their host genotype. But we actually didn't see a difference. And so that sort of suggests to us that maybe the actual genotype of their human cells is not the major factor. And so maybe it's due to sort of shared environment early in life or a variety of other different possible things. You can also ask questions of this type of data of just whether or not there's shared bacteria that are found across all people. And so sort of surprisingly, we found after looking at this data that there's no single abundant bacterial species that we always see in everyone's gut. So there's a lot of diversity between individuals. If you just look for bacterial species that are found in the majority of people, you find a few, but really nothing that's found in all. Okay. Additionally, you can start to sort of tease apart, whether or not there's specific bacterial pathways or genes that are found more abundantly in obese versus lean people. And so we're starting to get at that and hopefully now start looking at that in more detail. But sort of intriguingly, a lot of these genes that were found more abundantly in obese people were related to carbohydrate metabolism. And so, maybe certain communities might have an increased ability to harvest energy from the diet, and this could actually provide more accessible calories for the host. Okay, so the final thing I want to tell you about is just experiments that we've been doing in what we call humanized mice. And so this is sort of a strange and sort of fun experiment where we, again, start with these germ-free adult recipients, but then we wanted to ask whether or not we could reconstitute a human community into mice. And that would allow us to do a lot of experiments that we can't do on humans, but still looking at the same microbes that we would normally find in a human that we study. And so again, the reason we were interested in this is that it's very hard to study humans, so you can't really force people to go on a lot of different diets that you might be interested in. We don't have any germ-free humans, so you can't control what bacteria are in these humans. And it's even hard to just sort of monitor what people eat. And so these mouse experiments will hopefully allow us to get at that. So the first sort of set of experiments that we did was just designed to figure out whether or not we could transfer these microbes into mice. And so we took different mice that were raised germ-free and then colonized them with a sample taken from a human donor. And then we just followed these mice over time. And some mice were kept on a normal low-fat diet. Some mice were switched to a Western diet. And so the first thing we wanted to check was just whether or not we got a lot of the bacteria that were in the original sample into these mice. And it turned out to work quite well. So we got all of the major groups and then most of the sort of specific groups of bacteria into our mice. And that really allowed us to start asking different questions related to diet. I'll also tell you a little bit about how the community assembles in really young animals and how things vary along the length of the gut. And so the first thing we looked at was this effect of a diet shift. So we'd shown before this that shifting to this Western diet changes the composition of the community, but we didn't really have any idea of how fast this was happening and what the dynamics are. And so we use 16S sequencing to sort of look at the composition of the community over time. And this is just a clustering sort of graph. The important thing is that each sort of box is a different time point. So this is 1 day after we originally colonized the mice. And then the sort of how close each dot represents a different community taken from a different mouse. And then so the closer they are, the more similar the communities are to each other. So at the initial time point, they're all fed the low-fat diet, and then we can look at them a week later and then a month later. And so at a month after colonization, they're all sort of looking similar to each other. But now we can switch half of these mice to the Western diet and follow them over time and see how quickly we see a change. And so we're really surprised that after only a single day on the Western diet, we start to see all these mice that are fed this altered diet shifting over to the right. And then after a week and then following them for 2 months, it goes a little farther and then sort of stabilizes.
One time, go back. What are those changes when it moves to the right? What is that? That's more of what?
Yeah, so I think the easiest thing this is sort of a clustering-based approach, but I think the much more intuitive thing is on the next slide, where here we're looking at these major taxa or major groups of bacteria that are in the gut.
So changes in the bacteria in the guts.
Right. Yeah. So the Bacteroidetes are these blue bars. The Clostridia are the red bars. The more minor or normally minor groups like the bacilli or Erysipelotrichi or Proteobacteria are these other colors. And we can just sort of look at how this changes over time. So the original human donor looks pretty similar to these mice over the first month. And then if you follow these mice out over the next 2 months, just staying on the low-fat diet, which is this group of bars, they sort of stay around 50-50, so mostly Clostridia, mostly Bacteroidetes. But then after switching these mice to the Western diet, we immediately after 1 day see an increase in the Erysipelotrichi, which are one group within the firmicutes. And then after a week, we start to see a rise in bacilli. So really these large taxonomic groups of bacteria that shift in response to changing the diet.
Is there any, have you found out any difference in how they contribute metabolically, the Bacteroidetes versus the firmicutes? Like, do they the metabolism that they carry out in the intestine, do they preferentially do something versus something else?
Yeah, that's a great question, and that's exactly the type of research that we're doing now is to, these are really broad groups that I'm showing here. So each group is actually composed of hundreds or maybe thousands of different types of bacteria. And so we're trying to tease apart, what exactly each member of that group is doing and sort of the functional relevance of an increase in the abundance of one particular type of bacteria.
On a break, what percentage of the bacteria that we eat actually survives its way down to hang out with the rest of these guys? There's a big trend now to eat in flora. You can buy little pills and here's your bacterial flora, and there's all the yogurts and all that stuff. I mean, how much of that I mean, could somebody turn around and say, okay, I'm gonna eat a Western diet and I want McDonald's every day and I'm gonna find some, nice tasty bacteria? I mean, does that even make any sense?
Yeah, that's a great question, and that's really one of the directions that people are headed to try to manipulate this community. So there are already probiotics that people take through, I'm sure you know Dan Active and Activia, the Dannon brands. Those bacteria can make their way to the distal gut, but they don't actually typically stay in the body. So it's sort of a transient thing where we're eating the yogurt every day. They might stick around for a few hours and then they're sort of flushed out of the system. And so there's a lot of interest in sort of understanding how those, how the doses of bacteria that we take through the food manipulate the normal residents of the community and, sort of what they do during their passage. But you might also imagine, there's a really great example from a paper looking at a bacteria that grows on sushi, where they found that there's a particular gene that allows this bacteria to break down the carbohydrates that make up seaweed. Sorry, it normally grows on seaweed, not on sushi. But the cool thing was that then they looked at the microbiomes of Japanese and American people, and they found that these genes for breaking down seaweed were found in the normal members of the community in these Japanese individuals. And so sort of another level of ways that you might not actually be transferring the bug, but you might be transferring part of its genome into other resident genomes that are in your gut.
How do you know it's not eat a Western diet and take a lean diet probiotic pill?
Right. Yeah, that's what we do. Yeah, we'd love to get there. Yeah.
So the sushi bacteria, how do you know it's only that gene that's been put into another bug and not that particular bacteria?
Yeah, so it's actually quite interesting. So because it looks, at least it looks, there's not sort of definitive proof for this yet, but what it looks like is that sort of one chunk of the genome of the seaweed bacterium was transferred into a much larger genome of a normal resident of the gut. And so this normal resident is found in many different individuals in many different ethnicities, many different countries. But there's something called lateral gene transfer where bacteria can sort of swap out different parts of their genome. And at least so far, it looks like that might have been what happened in this case. And so the hypothesis, anyway, is that you might be, ingesting these live bacteria, and then they might be transferring genes while they're on their way through. I just want to tell you a little bit more about a couple more studies we did, but I won't go into too much of the gory detail. We basically wanted to just ask the general question of, whether or not the diet of the donor mouse or the recipient mouse was more important. And the result was really, regardless of where the community came from, it was really rapidly shaped by the donor. The other interesting thing is that even in these humanized mice that are fed the Western diet or the low-fat diet, we see this difference in the response of germ-free mice. So you can sort of transmit part of this obesity phenotype through these different communities. We next sort of turned to an older question about the microbiota, which is just sort of how do these bacteria colonize really early in life? And so the benefit of these mice is that we can take samples from their very first, the very first time they make a pellet, and then follow them throughout these really early times in their life. And so we're able to show, again, this is sort of a rarefaction type plot where the curve is an indicator of how diverse the community is. And so at 14 days after birth, it's a very simple community of only maybe 30 or 40 different types of bacteria. And then this gets more and more diverse over time. And then also there's really dramatic changes in the bacteria that are found in the gut. So at these early time points, these purple bars are things like proteobacteria or like E. coli, for example, is one. Bacteria that can grow, a lot of them can grow very well in the presence of oxygen, which is not normally found in the adult gut. And so these very different types of communities that sort of resemble what have been seen in human infants And then as soon as you switch these mice onto solid food, they're immediately taken over by Bacteroidetes and these sort of regular residents of the adult community. We also took these mice and did another experiment that you can't really do in humans, which is to take the entire length of the gastrointestinal tract. So we go from the stomach all the way down through the small intestine, the cecum, which is in mice a very large outpouching that sticks out from the colon, and then fecal samples, and we can really look along every little section of the gut and see how the community changes. And this again is a clustering type diagram. The main thing just to point out from this is that all the dots on the left are taken from mice fed a low-fat diet from many different locations, and then on the right are samples from the Western diet. So regardless of where you are in the gut, there's sort of this split by diet. And then we can also just go through in terms of taxonomy and look, if we start at the stomach and then move through the small intestine and then the distal gut into the feces, sort of how do the populations compare? And hopefully it's clear that we saw in the small intestine it's really dominated by Clostridia, but then as soon as you step into the distal gut, things shift pretty dramatically, where the Bacteroidetes do much better. And so there's sort of these patterns along the length of the gut as well as between different individuals and animals that are fed different diets. Okay. And the final sort of thing I just want to point out in this, we're starting to sort of move beyond just describing which bacteria are in the community to really looking at their genes, but also their transcripts. And so looking at transcripts really tells us which genes are turned on in a given situation. And I think will allow us to get at function much more easily. And so we looked at these mice again. This is just a clustering comparing mice in the low-fat diet versus the Western diet. So based on gene content, you can sort of divide mice based on the diet they were on. You can also divide them looking at gene expression. So this is the expression of genes just from one type of bacteria that's more abundant on the Western diet. And then you can also sort of parse out genes that are highly expressed on the Western diet across the entire community. And then you get relatively long lists. So we found just in this one study about 70 different genes in this one isolate that are differentially expressed. So genes that were more active on the Western diet or more active on the low-fat diet. And then we have a list of 500 or more different genes that across the entire community that are changing in expression. So, but hopefully starting to tease apart the different, sort of the most differentially expressed genes will help us sort of understand how this community responds to a change in diet. Okay. But the end sort of idea that we're sort of moving to is that, maybe we should think about not only the human genome, but our gut microbiome and how that plays into diet and many things like physical activity and metabolic balance. And so trying to gain more of a comprehensive picture. So we now take our entire metagenome into consideration. And this sort of, maybe moves us towards a model where we have our input diet, which, could change from day to day. And then the two sort of aspects of, either our own human genes that are helping us metabolize this diet, or microbial metabolism, and then how these end products of metabolism sort of work together to make us who we are. And I just want to end with a Kathy cartoon where, we at least have one supporter out there, which is Kathy. and just maybe raise the idea that, I'm not trying to convince you that gut microbes are the, even a major driver of obesity, but just make you think that maybe obesity is a little bit more complicated than only diet and only exercise. And so Kathy's friend says that she read The Fat Gene Makes Me Overweight, and she says, no, she already tried that and it only worked for a while. And then her other friend says, the stress hormone makes me overindulge, and she says, ho-hum, been there, believe that. And then her friend says, the overly efficient intestinal microbes in my system make me gain while others lose. And Kathy says, we found one that we can really stick with this time. Goodbye, miracle diets. Hello, miracle excuses. So, So with that, I'll just end by thanking my former thesis advisor, Jeffrey Gordon in St. Louis, and the many different people in his lab that contributed to these different studies. Now my current lab that I just started as a Bauer Fellow is at the Center for Systems Biology, just right next door. And that's my website. So it's been a real pleasure to be here, and I'd love to take more questions.
Have you looked at what's the fate of those, you said those short chain like fatty acids? I mean, do they get converted? I'm thinking of like, hardening the arteries or arterial sclerosis.
Yeah, it's really interesting. So there's sort of there's a few major short chain fatty acids that are made, and so one is butyrate. And butyrate, a lot of people like butyrate. So butyrate's actually like it's the major source of energy for the epithelial cells that sort of line the gut in the distal gut. And so that provides them with the ability to basically renew. And there's some, well, I don't want to get into that, but there's some indication that maybe butyrate is a good thing to have in your gut. But the other short-chain fatty acids like acetate can actually, are regularly absorbed into circulation, and then the idea is that maybe those are converted into fat tissue. And so they can be sort of linked together into triglycerides and stored.
Were in the fat and lean diets, were the calories equivalent and just the form of the calories?
Yeah, so it's sort of a difference at two different levels. So we'd love to sort of tease apart, which one is really more important for driving this shift, because they're different in sort of at the macromolecular level. So they have more fat overall and more sugar, but then they also really differ in the particular types of sugar that are found in the diet. So the low-fat, what we call plant polysaccharide diet, has sort of less refined sugars, and then the Western diet has things like sucrose or maltodextrin that are sort of normally found in what's typically thought of as an unhealthy sort of Westernized diet. But we don't really know which particular components of the diet, or if it's just sort of the level of fat as a whole that drives some of these changes.
Any research on like H. pylori, and is that like considered more of a normal bacteria now than like a pathogen, or is it still, as far as like stomach ulcers?
Right, yeah, H. pylori is a really interesting example, and one of my good friends worked on that for his PhD. It has its pros and cons, I guess. It's found, I think the current numbers are maybe like 1 in every 2 people are colonized. It certainly has been linked to things like stomach ulcers, but also maybe to diseases like chronic atrophic gastritis and other sort of problems that you can have in your stomach, maybe even stomach cancer. But then there's also some data that maybe suggests that it could be protective for cancer of your esophagus. So it's not clear whether or not, sort of completely removing H. pylori is going to be better off, or if there's some benefit to actually having it in your system.
Any of the bacteria in the human gut involved like cows for breaking down cellulose that we could then benefit from or not benefit, as the case may be, in terms of?
Yeah, so they're really good at breaking down polysaccharides like cellulose. And then we think we've found some genes that look like they can actually break down cellulose. And in particular, we did a sort of a deeper analysis of just 2 twins. It was one monozygotic, so identical twins. And one of the twins had a huge sort of array of genes that form what we think might be forming what's called a cellulosome, which is a sort of complex structure that allows bacteria to break down cellulose. And then her twin didn't. So we don't know if one twin was eating pencils or or why that was the case. But yeah, it looks like maybe they can do that. I mean, obviously, we're not as good as cows, so we can't just eat grass. But But yeah.
You had mentioned before that a number of these bacteria seem to be able to bring in different genes and whatnot. So now I'm a little confused on how we identify, using this DNA sequencing, how we're identifying different species if they can be so varied.
Yeah, that's a great question. So the gene that's typically used is 16S, and so that basically gives us an idea of or we think it allows us to group these bacteria by their evolution. That's sort of a long-term, or relatively long-term view of similarity. But there's also what's called horizontal transfers. So as these bacteria are evolving, they're also sort of trading genes with bacteria that are not on the same, and not necessarily, they're close neighbors on the evolutionary tree. And so that complicates things a lot for bacteria. So, unlike in larger animals where you're not typically throwing a human gene into a corn or something else, these bacteria, can transfer genes across relatively large distances.
Well, we typically in my high school biology class teach a lot about enzymes from the pancreas, for example, that that do digestion, and bacteria help out. But obviously it's a much bigger role. Is there any way to put a number on that, of contribution of bacteria compared to enzymes, or?
Right. No, that's a good question, and it's not an easy one to answer. There's some older estimates that, maybe on certain diets that might be found in Europe, for example, you might be able to place the estimated amount of calories as somewhere around 10%. But I think it's gonna, it's still early days, and so I think there's a lot more that we need to study before we can really say, given a certain diet, which components are going to be modified by the microbiota, and then how much of a role they're going to actually play. So the sort of the long-term goal is that we're going to be able to, predict not only the impact that the diet will have on these bacteria, but also, predict how much of a role they're going to play in breaking down a given set of substrates. But I think there's a lot more to learn before we can really, predict that well.
So you were looking at just healthy individuals or healthy mice. Have you looked at things like Crohn's disease or other digestive disorders?
Yeah, I haven't myself, but there's a lot of research, like Curtis mentioned earlier, as part of this Human Microbiome project. They're looking at, not as part of that one, but as part of another European microbiome project. They're looking at Crohn's disease and ulcerative colitis. So there's a lot of interest in the microbial role there. There's also interest in the role that the microbiota might play in changing how we respond to a drug that we might take, or how different communities respond to an antibiotic, and then other diseases are of interest too. So there's sort of a wide panel of things that are being studied. There's no particular bacteria that's been shown to actually cause something like IBD. But there are some really interesting studies, I think, that are coming out of mice. For example, there was a recent one where they showed that if you have a certain genetic predisposition to Crohn's disease due to this gene that was identified as part of a human study, it's an autophagy gene, but mice that are sort of predisposed to Crohn's disease and then are hit with colonization with a certain virus, actually develop symptoms that are similar to what we see in people. So maybe there's a combination of different things going on, where sort of the presence of bacteria and maybe different viruses, and then also your own genotype could lead to IBD. But it's obviously a very complicated picture that's coming out of it. Yeah.
I was wondering if there was any evidence that our flora make us prefer one sort of food over another. Is there any feedback?
Yeah, there's this paper from a guy in the UK that looked at chocolate consumption. I don't really know what to make of it, but it was interesting. They took you can take urine samples and sort of profile the metabolites that are there in sort of comprehensive way, just like we would if we're looking for bacteria. And so they found that like a certain collection of microbes could be used to, or a certain collection of metabolites could be used to predict whether or not the people would eat chocolate. But I don't know.
How about when you talk about people with Crohn's disease? How about trying to change the flora of a person with Crohn's disease to cure it?
Yeah, so not necessarily for Crohn's, but there's the C. diff associated colitis, which you might have heard about. There's a paper in the New York Times or an article in the New York Times a few weeks ago, or maybe a few months ago now, but very interesting as sort of a last line of assault against these really severe problems with colitis. They took a married couple and where the wife was suffering from colitis, and they gave her, took a sample from her husband, a fecal sample, and then used that to colonize her gut. And that resolved some of the symptoms of colitis. So sort of the extreme microbiota transplantation in humans now, yeah. Okay.
Thank you.